Hello, everyone. Today's focus is going to be on thalassemia, a complex blood disorder that affects thousands of people around the globe. This condition involves the genetic makeup of hemoglobin, the oxygen-carrying component of our blood, leading to an array of health challenges. Before we tackle thalassemia, it's crucial to understand the basics of hemoglobin. Hemoglobin molecules are comprised of 4 globin chains, each holding a heme group. During different stages of human development, these chains vary. In early fetal life, embryonic hemoglobins such as Portland and Gower are present. By late fetal life, fetal hemoglobin, or HbF, takes the lead. HbF uniquely adapts to capture oxygen from the mother's blood, a critical function before birth. After birth, a switch occurs, usually between 3 to 6 months, transitioning to adult hemoglobin, HbA, while HbF levels decrease but remain detectable throughout life. These disorders, especially thalassemia and sickle cell disease, are prevalent in regions formerly or currently affected by malaria, as carriers of these conditions have some resistance to the malaria parasite. Hemoglobin disorders fall into 4 main categories. 1. Disorders affecting globin chain synthesis, thalassemia being a prime example. 2. Structural defects like sickle cell anemia. 3. Rare unstable hemoglobins. 4. Very rare conditions leading to increased or abnormal hemoglobin types. These thalassemia disorders are divided based on the affected globin chain, alpha, or beta. This condition arises from issues with the alpha globin genes on chromosome 16. The severity depends on how many genes are affected. Hydrops fetalis, all 4 genes are inactive, leading to fatal outcomes either in utero or shortly after birth. Hemoglobin H disease, 3 genes are inactive, resulting in severe microcytic, hypochromic anemia, and spleen enlargement, among other symptoms. Alpha thalassemia trait, 1 or 2 genes are affected, often causing mild anemia and high red cell counts. Beta thalassemia ranges from severe to mild, depending on the mutation affecting the beta globin gene. Thalassemia major, this severe form manifests early in life when the normal switch from gamma to beta chains should occur. Symptoms include severe anemia, growth delays, and organ complications due to iron overload from frequent blood transfusions. Thalassemia intermedia, this less severe form might not require regular transfusions and presents later in life. Beta thalassemia trait, this mild form involves slight anemia with normal iron levels. Diagnosis of thalassemia involves blood tests showing specific changes in red cells and unique hemoglobin patterns, confirmed by DNA analysis for precise genetic mutations. Management includes regular blood transfusions, iron chelation to prevent iron overload, and monitoring for organ damage. Advanced treatments include stem cell transplantation, which can offer a cure in some cases. Given the genetic nature of thalassemia, prenatal screening is vital. If both parents are carriers, there's a significant risk for their children to inherit severe forms of the disease. Modern techniques like DNA testing of fetal cells allow early diagnosis and informed decisions by prospective parents. Thalassemia is a profound example of how genetics directly influence health. Through understanding and managing this condition, we can greatly improve the quality of life for those affected. Whether you're a healthcare professional, a student, or simply curious about genetics and health, I hope today's discussion is helpful. Whether you're a healthcare professional, a student, or simply curious about genetics and health, I hope today's discussion is helpful.